cell division. We'll start talking about cell division by looking at why cells divide. One reason cells divide is for growth. We all start off as one cell, and through cell division, we get to the point where we are today. The second reason cells divide is for cell replacement. Our body is constantly losing cells. They're either dying or being lost in other ways. So we need to replace them. So cell division helps us replace lost cells. Third reason for cell division is for wound healing or repair. So anytime a part of your body gets damaged, cell division will occur to repair that damaged area. The final reason for cell division is for reproduction. There's two different types of reproduction. We have asexual reproduction and sexual reproduction. Asexual reproduction, the cell makes an exact copy of itself, or the organism makes an exact copy of itself. And so that's one way to go through reproduction. The other is sexual reproduction. And so we have two cells that are formed that combine to form a new cell, which becomes a new organism. All cells have a life cycle, which we call the cell cycle. And as you can see in this diagram of the cell cycle, the majority of the time, the cell is in interphase stage of the cell cycle. There are three phases in the interphase. There's G1, S, and G2. G1 and G2 is when proteins are synthesized. That's when organelles are made. And then the cell is also growing. During the S phase, DNA replication occurs. So our chromosomes are usually a single strand, but during replication, it makes a copy of that strand, so now it becomes a two-stranded chromosome. And so each strand is an exact copy. And so when the cell divides, each cell will have the same copy of chromosomes. So after G1, S, G2, we then enter the M phase, and that's when mitosis or meiosis will occur, and cytokinesis. Mitosis or meiosis is when the nucleus divides. Cytokinesis is when the cell divides. So the first part of cell division that we'll look at is mitosis. And mitosis occurs when we have cell division for any of these three reasons. So we're going to be making exact copies of the original cell. So mitosis makes exact copies of the original cell. There are 4 steps to mitosis. We call it, or you can remember by saying PMAT, P for prophase, when the cell is preparing. So the chromosomes plump up, the nuclear membrane breaks down, the spindle fibers start to attach to the chromosomes. Then we have the M for metaphase, when the chromosomes line up in the middle. And then A for anaphase, when the chromatids are pulled apart. And then finally telophase, T for 2 or telophase. We now are going to have 2 nuclei forming. And again, this is just nucleus dividing, not the cell. But as you can see at telophase, the cell is getting ready to divide. The division of the cell is called cytokinesis. Cyto is another word for cell, so the cell is dividing. In an animal cell, it divides by cleavage furrows being formed, which pinches the cell into 2 new cells. In plant cells, a cell plate forms between the 2 nuclei, and that plate will then become a cell wall. So those are the 2 different ways a cell can divide during cytokinesis. And looking from start to finish, you see our mother cell and you see your daughter cell, and they are exactly the same. So again, mitosis starts with 1, we end with 2. The 2 daughter cells are exact copies of the mother with the same number of chromosomes. So chromosomes, review of a chromosome, their structure made of strands of DNA, molecule, and protein. So chromosomes come in different forms. This is what you usually see. It has the X shape. And what that chromosome has, how it's made up, is that there's one strand of chromosome, there's a copy of it. And we call that chromatid. So the two copies, they are chromatids. And they're held together in the center at a place called the centromere. The centromere holds the two chromatids together. So before DNA replication, the structure of a chromosome is just one thin strand. And we call that thin strand a chromatin. I think a thin and tin. Chromatin is just when it's one thin strand. After DNA replication, there's two strands, and we call each side of each strand in that structure a chromatid. So no longer chromatin. Chromatin is when it's only one chromatid. There are two chromatids in this chromosome. It's that one strand, but now it's stuck together at the centromere. And again, centromere holds the two chromatids together. Chromosomes have two different types. We have what the majority of our chromosomes are, which are autosomes. That's just about every chromosome in our cell. But we also have special chromosomes called our sex chromosomes. Those are the chromosomes that are going to determine if you're a female or a male. And they're your X and Y chromosomes. So now we're going to look at the type of cell division that happens for sexual reproduction, and that's meiosis. So meiosis, when we're talking about meiosis, is another thing. When we look at chromosomes, we talk about there being homologous pairs. So all of our chromosomes, we have two copies of each chromosome. There's a copy that we got from our mother and a copy that we got from our father. Those two together are called homologous. They're the same type of chromosomes. They have all the same genes on there, but one's from your mother, one's from your father. So you have two copies of all your chromosomes. During meiosis, those homologous pairs get separated. So we're splitting apart the mother and father chromosomes. So the number of chromosomes, we call them different names. So when we have the two copies, it's called diploid. So when we have the mother and father, it's diploid. When we break the mother and father pairs apart, those homologous pairs, then the cell is haploid. So we have half the amount as the original cell. So the phases of meiosis, same phases as mitosis, but the division occurs twice. So each of those phases are going to occur two times. And the first time, during meiosis 1, we just name the phase with a 1 after it. The second division is called mitosis 2, and we name the phases with 2 after it. And the same things are happening in each of these phases with a little bit of a difference. We'll talk about that. So this is where the difference is. In meiosis 1, what happens is those pairs line up together. So the pairs are together in the middle. It's not just one line of all the chromosomes, but we have pairs lining up in the middle. And then those pairs separate from one another. So this is where we separate the mother and the father chromosomes. And an important thing that happens during prophase 1, when these chromosomes are lining up or pairing up, sometimes they cross over and they'll exchange parts of their chromosome. And so this is called crossing over, when a part of one chromosome gets switched with the other chromosome. And this gives us some variation in the end result of the final cell. It's not going to be exactly like the original cell, and this is one way it's not the same. Okay, so meiosis 1, our pairs separate, and then meiosis 2, now you can see they're lining up in a single file, and this is when the chromatids separate from one another. And so we had two cells that were formed during meiosis 1, and both of those cells go through prophase, metaphase, anaphase, telophase, and at the end we have 4 cells. So again, start with mother cell, go through first stage of meiosis, which is meiosis 1. We end up with 2 cells, and again you can see the pairs are lining up in the middle. And then during meiosis 2, there's a single file of chromosomes in the middle, and by the end we have 4 cells. And those cells at the end of meiosis, we call those gametes. So if we take a quick comparison, mother cell, mitosis ends with 2 daughter cells that are exact copies. At the end of meiosis, we have 4 cells that are not the exact copies.